Okay, very good. Thank you. So welcome everybody. It's 12:30 and we're going to get started with our webinar. And so I'm Laura Burris, I'm the chair of biology, and I'm really happy to be here today hosting this event along with Jose de la Torre, the associate chair of biology. And today we've collected 8 amazing panelists to talk with you about sciencing up about COVID-19. And I want to say that this event is really in response to requests by students saying, hey, we're in a biology department, and why aren't we talking more about the science of COVID as a community? And so I put out a call for people who would be interested in talking about it, and we got 8 people, just boom, boom, boom. So I'm really pleased to be able to do this. And I think what I want to emphasize about all of our panelists is that, like you, none of them are experts in COVID-19. Nobody here, that's their research specialty that they studied through graduate school. We're just a bunch of scientists who are sciencing up about thinking about things. And so now more than ever, we really need to think about our community health in terms of using evidence-based data. And I hope that these 8 panelists will really model to you how to do that, and then we'll have some time for Q&A. And so the format for today is going to be that we're going to have introductions right now. We're going to start our first panel session with Pluni Pennings, Ed Connor, Leti Marquez Magana, and Cristina Morales. And then we'll have a Q&A for about 20 minutes, and then we'll do a second panel session where Kaho Tistammer, Ravinder Sehgal, Erica Sanchez, and Mike Goldman will speak, and then we'll have another Q&A. And so we'd like you to ask that you hold your questions. You'll be able to type them into the Q&A panel, but hold your questions until we get to those parts. So I think to give us the panelists as much time as they can and as much time for Q&A, we'll go ahead and start with Pluni Pennings, who is a recently promoted associate professor of biology at San Francisco State. So I'm going to stop sharing and let Pluni start sharing.
Hi, everyone. It's great to see so many of you joining us here today. As Laura said, my name is Pluni Pennings. I think I'm officially still an assistant professor, but soon I will be able to call myself associate professor of the biology department at San Francisco State. My regular research is on HIV and drug resistance. So I'm used to thinking about viruses, but never studied viruses before. And I'm going to share my screen. And the topic of my short presentation today is how many people do we think have actually had COVID-19? And so we have official numbers of people who have tested positive for COVID-19 or for carrying the virus, SARS-CoV-2. And so the official numbers are roughly 1,400 people in San Francisco, a little over 7,000 in the Bay Area, almost 1 million now in the United States. It's really incredible, and 3 million in the world. But even though these are high numbers, especially for the 1 million for the United States, probably the real numbers are still much higher. So I actually wanted to do a poll, and I'm not sure if how this is gonna work exactly, but I'm curious. Laura has already started the poll, I think.
Oh.
Is it open?
Yeah, it's open.
Okay, so the question is, have you had COVID-19? So everybody who's here, we have about 200 participants, so that's great. That's a great sample. So please let us know, yes, you tested positive for the virus. You think you had it, but you haven't been tested. Or you're like, maybe, but maybe not. Or you're pretty sure that you never had it. And I think Laura can tell me when most people have voted.
Yeah, we're up to almost 90%. So I'm going to go ahead and close up the poll, and then I will share the results.
Perfect. Okay, so nobody, out of more than 200 people who are here, nobody has actually tested positive for COVID-19. But quite a few, 7%, think they've had it, but they haven't been tested. Some people say like, maybe yeah, maybe no. And the majority actually says that they have not had it. Well, thank you very much. One of the things that we know from testing let me go back here, from testing in UCSF is that of all the people who go there to get tested, only about 5% actually test positive. So even if you think you've had it, it doesn't mean that you've actually had it. So what are the real numbers? So we don't know, and we will never know for sure, but seroprevalence studies can really help. Seroprevalence studies look at whether or not you have antibodies against the virus, against COVID-19. So there have been several coming out recently. One of them came from here locally from Santa Clara County. I think it was done poorly and we cannot trust these data. But another one was done in New York State and New York City. And I think that was a much better study with a more random sample. And they estimate that seroprevalence in New York City is 21%. So 21% of people recruited at a supermarket had antibodies. And that would mean that 1.8 million people just in New York City have had COVID-19. The confirmed number of cases in New York City is 141,000. And so that suggests that only 1 in 12 is detected. We also know that at the time that that study was done, 14,600 people had died of COVID-19 in New York. And so that suggests an infection fatality rate of 0.8%. So out of all the people who have had the infection, 0.8% have died in New York City. And so that's likely 20 times higher than the flu. So it's really much worse than the flu. If the infection fatality rate is the same in San Francisco, that suggests that about 2,800 people have had COVID-19 in San Francisco. So that number is much, much smaller than in New York, even given that our city is smaller than New York. And about 32,000 people in the Bay Area. So in conclusion, the real numbers are really high. If only 1 in 12 is detected, it may mean that we have 12 million people in the United States who've had this disease, which is really incredible for a disease that, you know, a couple of months ago, nobody had heard of. It was non-existent in this country. The number of cases in the Bay Area and in San Francisco is really quite low compared to other places. And that is probably thanks to very early and very strict stay-at-home orders, and thanks to some real leadership from people in power, such as our mayor in San Francisco and Dr. Sarah Cody, who is a public health officer in Santa Clara County. So thank you. I'll be happy to answer questions later on during the Q&A.
Okay, thank you, Pluni. So if you'll stop sharing your screen, our next speaker or panelist will be Ed Connor. We'll talk about COVID-19 by the numbers.
Hi, welcome everybody. I'm going to talk about some really dry, esoteric sort of statistical modeling techniques. So you might want to take a break right now and run to the bathroom or whatever you need to do. So as part of my class in advanced quantitative methods called advanced biometry, we decided to try to look at some of the data that's been accumulating about the number of confirmed cases and the number of deaths from COVID-19 throughout the United States. So we were able to download data set from the Johns Hopkins site, which provide information mostly on a by county basis. And there are almost a little bit more than 3,000 counties in the US, but only about 2,700 and so were actually reporting data for the Johns Hopkins website. And so we assembled that data and tried to locate as many sort of potential explanatory variables or covariates to add to that data set. So we wanted to find information about poverty rates, median income levels, population density, the information about whether a particular county had high rates of mortality from respiratory diseases or heart disease, so other kind of pre-existing medical conditions, to see if any of those things that help explain variation broadly throughout the country in the number of confirmed cases or the number of deaths. And so we immediately, as because of what we focus on in this class, go beyond sort of classical statistical modeling and use techniques that allow us to relax assumptions that have to be fairly rigidly adhered to in classical modeling. So we could fit models to data that more appropriately work with the natural sort of scale of that data. For example, the data I've talked about is mostly count data. And count data have to all consist of positive integers. So a normal distribution consists of both negative and positive values, or can, and it consists of real numbers. So they're not just integer values. So it's probably more natural to model count data as Poisson data, where you can only have positive integers. So we fit a variety of models, used a variety of techniques, hierarchical models, additive models, hierarchical additive models, and various kinds of things that have come out of machine learning, such as regression tree type models, random forest, and boosted trees. And so these are all topics that we had covered over the course of the semester in the class. And so the students in the class were familiar with these, but hadn't sort of say, let's attack one problem and try to apply a variety of techniques to see how we can do and try to explain some of this variation. It was first a challenge to put the data set together because as you'd think there would be a lot of convenient data sets that would have this information, but they often are not that convenient to actually get access to. Either they're not at the county level, some things are at the census tract level, some are at the state level. So it's difficult to do everything we wanted to do because it took a good bit of work to put the data set together. In general, though, we found that fitting hierarchical models, Poisson models, that were corrected for overdispersion in a lot of technical terms that my students have learned about in the course of the semester. Those models, which included population density, the number of people tested, and 3 other covariates that had to do with income and poverty levels, as well as medical conditions, could explain about 6% of the variation across the country, from county to county, in the number of confirmed cases, and about 33% in the variation in the number of deaths. Regression trees yielded very inferior results, although we tried both random forests as well as boosted trees.
1 minute, Ed.
So anyway, that's the sort of exercise we've engaged in to try to learn a little bit about what we could imagine from what we could from the data. There may be other covariates that you might think of that we should have included, that we haven't yet included, but we could still do some additional work to see if we can find that data and include it in our data set. So please feel free to ask questions later during the Q&A period.
Thank you, Ed. Next up is Leti Marquez Magana.
Hello, I am a professor of biology, and as was said, I'm not a COVID expert. Nonetheless, I am the director of the health. So here's my information. I go by she, her, Aya, and I'm the director of the Health Equity Research Lab. And just everybody nod if you see the screen. I just want to make sure. Okay, good. And so nonetheless, we have just been recently funded to undertake a study nationally with scholars across the country to gather information that'll better increase understanding about the data, the population level data that Ed Connor was talking about in terms of race and ethnicity distribution. So stay tuned. But with respect to this title, Non-Random Distribution of Health, I wanted to go over how we think of health in the health disparity field. And so this is a picture of the social determinants of health. But most folks from high school on, you've learned about gene-environment interactions. So it's never just about the genes. It's never just about the environment. It's the interaction in terms of getting, for example, health. So health is at the base of this visual. And you can see that health interacts with a number of things in terms of the genetic and other biological factors, but it really is a small component of our overall health. The health is dictated by our behaviors. Do we go for walks? Do we eat healthy? That sort of thing. Do we have access to medical care? And the thing is those are individual level, but they're informed by our living and working conditions. So if you live in a community where walking is a good idea, it's not dangerous, you may do it. If you work at Target these days and you're not allowed to wear a mask, you can't change your behavior. You're more apt to get exposed. And then you can't go to see the doctor if you can't get the time off. So that is all framed by economic and social opportunities and resources. And so in this case, social disadvantage leads to a non-random distribution of these determinants that could be helpful. And what happens at the biological level is that there's a chronic stress, so that leads to cortisol dysregulation. What we study in the Health Equity Research Lab is telomere shortening, but it also increases your inflammatory responses and lower immune response. So you can see that if this chronic stress is happening and you're in a community where you're experiencing these things, you're set up to, done all the reading, to have greater incidence of exposure to COVID and then worser outcomes. So we know that, in fact, that's coming to bear. And so here's the statistics. And if you do your Twitter account, hashtag we need to do better. A lot of this information is appearing. But in Michigan, for example, African Americans are 14% of the population and 41% of the deaths. In Louisiana, they're 32% of the population and 70% of the deaths. And from my colleagues at UCSF, they're telling me that the ICU beds at the county hospital are full of Spanish-speaking Latinx. In fact, my friend Tomas Aragon, who's our health officer, has said to me, we need tremendous assistance with the Latinx community, especially those who speak Spanish. So yesterday we were talking about some of the contact tracing and that sort of thing, and the need to have individuals with the community cultural wealth to actually navigate these environments and get the better numbers and mitigate some of what's going on. So what can we do? So I think, first of all, as scientists, we need to have courageous conversations. We have to recognize that these disparities existed pre-COVID and they will exist afterwards unless we actually make some changes. Jason Corburn at the UC Berkeley School of Public Health, and I show, there's a link to the talk, talks about the fact that right now the decisions that are being made don't include people who have power and privilege. I know I have one more minute. So, and so these are some of the things that we need to think about. But in the end, what we're doing in Biology 331, for example, is we're spreading accurate scientific information in culture and in language. So here's an Instagram that was created by Gabriela Acevedo, Alvarez Acevedo, who's actually a master's student in our department. And it's explains some of the concepts that Pluni put out on her video. We need to talk about the fact that race has no genetic or scientific basis. That was announced at the when the Human Genome Project was theoretically completed, and yet folks still continue to talk about that as why some folks have worse outcomes, and yet it's not biological. And so there are opportunities to do some contact tracing. Those employment opportunities are coming out, and contribute data stories. So this is the health atlas that UCSF has put out. They're asking folks to actually include some of their own data in there in order to make that a richer resource. It's right now built from the LA Times, which actually has a lot of data sources for doing your own analyses. And this summer, we're going to be working with scholars at San Francisco State and across the country to amass great amounts of data. Sorry. Thank you.
Okay. Thank you, Letty. And we will have these slides posted, I think, after the talk, so you'll be able to get those links out of the slides as well. Next up is Cristina Morales from the California Department of Public Health, and she doesn't have a video, but she will have some slides to share. Christina, are you here? Can't hear. Okay, here we go. Got screen sharing. I cannot hear Christina.
Hello?
Oh, there I can.
How's
There
that?
you go. Thank you.
So thanks to Dr. Burris for inviting me to present today, and to my husband, Dr. Julio Ramirez, for volunteering me. Hopefully today will be somewhat interesting. So as Dr. Burris mentioned, I work at the California Department of Public Health in the Viral and Rickettsial Disease Laboratory Program, specifically the Respiratory and Gastroenteric Diseases section. And our lab has been directly involved in testing for SARS-CoV-2. So what I want to show you today is kind of like a timeline of our testing as we've gone through since the beginning. So before I get started, as talks shift to ending shelter in place and reopening businesses, one of the keys to California's doing so is the expansion of testing for this virus, and to see who has active infections so that we can do things like seeing who has active infections and the contact tracing and testing of the folks that came into contact with these individuals and monitoring them for the virus as well. Unfortunately, not everyone who wanted to be tested has been tested. So why, one of the questions, big questions out there right now is why is that? So as many of you know, for testing for SARS-CoV-2, it's a multi-step process. Of course, you've heard that it involves real-time PCR, real-time RT-PCR. However, before PCR can happen, you have to extract the nucleic acids from the specimens. And since SARS-CoV-2 is an RNA virus, you have to take extreme care to make sure that the RNA is not degraded and therefore give you false negatives. Then even before that, you have to collect the specimens from individuals, typically using a synthetic swab, usually using nasopharyngeal or oropharyngeal collection. Now, back in February, when the demand for testing was low in California and the United States, we were already making preparations to ramp up. And as you can see from these numbers here, I gave a presentation to our group of 50 on February 19th. The United States only had 15 confirmed cases in these states, and California only had 8 confirmed cases. As such, you can see that the testing materials were in pretty good supply for all of these. There was, of course, an issue with the primers and probes from CDC, resulting in a low shortage of supply for that primers and probes. But eventually, this supply was pretty good. So this was back in February of 2020. Hard to believe that that was only a short 2 months ago. So as we moved into March, many public health labs still did not have the CDC primer and probes, but luckily we were able to acquire that at the state lab. The California Department of Public Health was one of the first to get the fixed primers and probes, so we ramped up our testing. However, many of the other 24 county labs, so a lot of the testing was still going on in the public health labs, could not start their testing because they didn't have this very important reagent. Eventually, CDC partnered with a commercial company, IDT, to produce enough of those primers and probes. And by the end of March, all the public health labs in California that were capable of doing this testing had enough of those primers and probes, as well as the enzyme, this TAF enzyme kit, to be able to start doing testing.
Got 1 minute.
Unfortunately, now that higher volumes were happening, there were low levels of the extraction kit available, so that caused a backlog in the testing as well. So come to March, a number of other extraction kits and platforms came online. Some were 8 to 96 specimens. There was still a huge backlog, though, as the number of samples and specimens requested came online. So there was still this backlog, and also what was happening, we had a backlog in the number of swabs that were able to be used. So hopefully what is happening into May is we won't have that backlog anymore, and that so we can ramp up our testing to a good amount. What we're currently seeing is that we can do about 25,000 tests per day in California. The goal is to have 100,000 tests per day and being able to test asymptomatic as well as symptomatic patients. Is that possible? This is what we've done at CDPH to ramp up our testing, but there are several key questions. Will the supplies and materials keep up with demand? And how many people will be able to be tested and when? So those are a lot of questions that still remain unanswered.
All right, thank you, Christina. So now that we've had our first 4 panelists, I think we're doing really well at staying on time here. I'd like to open it up for questions and answers now. And so in your Q&A box, please start typing in some questions, and Jose de la Torre and I will start to kind of dive in on those. And so can the panelists see the questions too?
It's in a special, it's in a special
Q&A box.
Yeah.
Okay, well, I want to start off with one for Christina. So you say that we should be able to ramp up to 100,000 tests per day in California. what's the number? The governor has one of his 6 criteria for kind of lifting sheltering in place being about testing. What is his goal? Do you know?
That has been what we're told the governor's goal is to try to test up to 100,000 per day. It's a big question of whether we'll reach that though.
Okay.
So that's what we've been mandated to do and try to get up to 100,000 statewide with all of the labs that are currently testing.
And how long do you think it'll take to get to 100,000?
Like I showed you in my last slide, we're currently at 25,000 tests per day, and that's including all the commercial labs, all the public health labs, all the non-public health labs. We're ramping up. Our capacity at the Department of Public Health at the state lab was 150. Now we're building capacity to about 1,000. So 1,000 tests per day. If all the other labs can kind of ramp up in that way, maybe we could reach it, but there's also a lot of other labs that are coming online as well.
Okay, thank you. Letty or Christina, can you address mobile COVID testing in minority communities? The person who wrote this finds only 1 mobile unit for the entire Bay Area that's not specifically focused on minority communities. Do you have any information about that?
So I don't know if Christina knows about mobile units. What I do know is that there's a lot of distrust and mistrust by the communities. So folks, because of historical transgressions in terms of what scientists and physicians have done, they're more afraid of going to get tested than just staying at home and taking care of themselves. Oh, I thought I was, okay, I am unmuted.
No, you're not, you're good.
Okay, good. So I do know that they're setting things up in the Mission right now.
Okay, very good. And so how would people find out where to get tested? Do you know?
What I did see on ABC News on your side is that Luz Pena has been covering what's going on in that community. And so if you go to ABC News, you know, there's a little consumer site with respect to that. And there was another question just about the comorbidities. I think that obviously the underlying conditions of folks is what is showing to be the case as to whether you die or not. And it's not clear what those are. But right now, elevated cytokine seems to be a big thing. So those are the inflammatory responses, and having things like high blood pressure, heart disease, and those diseases have disparate outcomes. So we know nationally that African Americans, Latinos, Native Americans have a lot of issues with health issues. So it is not surprising that what we're seeing is the unveiling of health disparities that have existed forever in terms of COVID-19 disparities.
Okay, thank you. Let's see here. I saw one that I wanted to chip off. I'll give you another one, Letty, while I'm at it. We've got a question about cases in the Navajo Nation and that they are disproportionately high as well, including in kind of areas around Phoenix and Albuquerque. So what is your view of cases in rural America, like in the Navajo Nation?
I think, unfortunately, we don't have enough data. I think that that's one of the goals for this summer project that we're launching, is to amass the data. And like the person that wrote this, that should be contributed to the UCSF Health Atlas. So that person has firsthand knowledge of what's going on in their community. And so it's not coming from the county, it's not coming from sources that people think are, you know, reputable, but at this point, anything is good, because no data, no policy changes. And so go to UCSF Health Atlas, you know, try to input some of this data, and together we can put together something that makes more sense for the whole country to move forward.
Mm-hmm. Thank you. In California, I think this one would be for Christina probably. What's the most accurate antibody test that's available right now?
So far, there's an FDA approved test. I don't know how accurate that is because we haven't started testing it in our lab yet. So right now, there's a lot of antibody tests that are out there. It's just a matter of it's an FDA approved test. And right now, there's only one approved one. So in terms of accuracy, I don't know.
Okay, thank you.
And it's important to distinguish the two components that go into accuracy, which is sensitivity and also and I'm just blanking on it. Sensitivity and specificity. Thank you, Pluni. So specificity is whether it detects other coronaviruses or other antibodies to other things. And sensitivity is how accurately it can detect the antibodies once they're there.
So I have a question that might go towards Pluni or somebody else if you think it's more appropriate. But how does the COVID-19 compare to the infection rate of, say, Spanish flu in the past or other pandemic diseases?
That's a good question. I think it's not extraordinary. I think it spreads fairly. It has this sort of surprising thing that you can be infectious even before you're symptomatic. That makes it hard to control. But I think just in terms of the number of people that you're likely to infect if we would live our normal lives, it's not extraordinary. Is a combination of the lethality with nobody having had this disease before, so nobody has immunity, that makes it, and of course it just being a respiratory virus that has a fairly high R value, it spreads quickly just like the flu would. The combination of these things is what makes it so dangerous.
Thank you. So here's a question for Ed. One person wants to know, what implications for our behavior can we extrapolate from your class's models? You got to unmute, Ed.
I think I typed in and sent an answer out, but I'll also try to.
Oh, okay. I think others might want to hear that too.
So we weren't able to put together a covariate which would measure something about the extent to which people altered their behavior and when they altered their behavior by sheltering in place or social distancing. If we could get a measure on a county basis of some sort of how much cooperation that people in that county had with the social distancing sort of guidelines, then we might be able to make some statement about whether it affected number of confirmed cases or number of deaths. But we don't really have that information. And I don't know that it exists at this point. Possibly the GPS data or Bluetooth proximity data that the phone companies, if it was available on a county basis, maybe that would be a way to look to see how much you've reduced your movement on average in a county and use that as a covariate in the modeling. But based on what we have, I can't really say that there's any message for how we should change our behavior.
And I think we also have to recognize that behaviors are sometimes, again, affected by systemic problems. So there are folks in Target, in the fields, in 99-cent stores who want to maintain social distancing, who want to wear the masks, but they're being told that they have to provide their own protection. They don't have the money. They don't give an alternate workflow so that they can maintain social distancing. So I think it's not just about individual behaviors. We need to think about
structural.
Yeah.
Yeah. And so I don't know who this one's for, but are the number of new confirmed cases of COVID-19 per day increasing or decreasing? And I imagine that would depend on where you're at. But let's say in California, do you think they're increasing or decreasing at this point? Or you need the panelists?
California. So something in the range of 12 to 1,400 per day.
And is that going up or down, do you think, at this point?
It's been fairly stable for the last couple of weeks.
Okay, so the curve is kind of flat, but
some days a good bit lower, but it's been fairly stable for the last couple.
Okay.
I think actually all over the United States, it's fairly flat currently.
Okay.
But
even in New York?
In New York, it's going down.
Yeah. Mm-hmm.
Yeah, so that flat curve over the whole country could actually be hiding that, like in New York, it's going down, but it means that somewhere else it's going up.
That's right. Maybe in Atlanta, I would say.
And the other thing that would be super interesting is to disaggregate by racial ethnic identity, because I would suspect that in some communities it's going down and flattening, and others it's going up.
Yeah.
Because, yeah.
Yeah. So there's a lot of data to be pulled apart here, which, you know, we have really good access to a lot of data through the Johns Hopkins site and others. So people can go in and try to disaggregate some of that. So there's some other questions about whether you have any sense from your studies of whether the virus might be seasonal or what will happen when flu season hits, and what the predictions are for that. Anybody feel qualified to weigh in on any of that or have any ideas based on your evidence?
My brother Miguel is the boss for Sarah Cody, the county. Okay, so they're saying that they're expecting that will happen in the fall, that it's going to come back, and they're planning for it. So all I can say, it's hearsay from Sarah, from my brother, from who, you know, is the supervisor for Sarah.
Thank you. And so go ahead, Jose.
Oh, there's also been a couple questions asking about what we need to achieve in terms of testing and contact tracing as a state in order for us to get to a point where we would feel comfortable, or where the governor might feel comfortable getting us back to work. I don't know if that's something that Christina can talk about from the California State Department of Public Health's perspective.
That's more something that I would have to talk to our epidemiologists about, because they have to see a certain decrease in the number of cases happening, how many cases are, say, hospital cases are there sitting in the ICU, how many new people are coming in, things like that. So that's not something I can readily answer. That would be a better question for one of the epidemiologists here.
What I can tell you is that I was on a call with the San Francisco Department of Public Health yesterday. They were saying they need 150 contact tracers. So that's just for the county, city and county of San Francisco. Currently, the individuals that are doing that job are the folks like the librarians and the lawyers that are in shelter in place. They're being moved around. And so once the shelter in place is lifted, the librarians and the lawyers will go back to their jobs, and they're going to be hiring contact tracers. And they're probably going to be working with San Francisco State and some of our practicums to have 150. So the answer is 150 is what they need now by like June 1. That's what. So
we're talking about 150 jobs, right?
When the shelter in place is lifted, yeah. But what they're talking about in the interim is that we have MPH students and health education students that have to fulfill their practicum in order to like progress in their degree. And so they're going to be using those classes and those folks to do the contact tracing.
Okay, thank you.
But eventually this might be a good source of temporary jobs for many people as well.
Yeah, that's what I was emphasizing. So we have kind of two issues here, which is obviously the health effects of the pandemic, but also the unemployment effects of the pandemic, the economic effects. So I've got a question here about, you know, there's some CDC recommendations out about what's safe and what's not safe. To what extent can we trust the CDC right now, given that they're being so highly censored by a non-science-based government? Anybody want to bite on that? Flooney?
Wait, sorry, could you repeat that question? I was reading another question.
Okay, that's okay.
So I'll bite on it. I'll bite
okay.
it. So the thing is I think the American public is experiencing what many folks of color have always experienced, is a complete distrust of a lot of the official words. And so I don't know what to do. I mean, I think that's why we need to be agents of change. Those of us who understand this kind of thing, we need to be able to communicate to one another in ways that are understandable so that we can share the knowledge that we have collectively.
Thank you. Anybody else want to weigh in about how they think about, you know, what the CDC or somebody like that says and how much we can trust that given the current censorship? Okay, that's a tough question. Let's see. So based on data we have access to regarding the progression of the virus, what is the statistical modeling for say about California's peak of infection? Anybody?
I think
so some of these questions, go ahead, Flooney.
I think it was thought that the peak had already passed, although the last couple of days there have been a lot of new cases in LA County. And so I think we could, you know, you can make a model, but then if people change their behavior, or if like a mayor decides to open the beaches, or if something happens, then, you know, there's no model that can incorporate all of those things. So it looked like the peak had passed, but if we open up things too quickly without having tracing in place, it may peak again. There may be many peaks.
Yeah, there's going to be some definitely rolling hills. You know.
Yeah.
So here's one for Pluni, definitely. I've been under the impression that COVID-19 is evolving rapidly. Looking at the nextstrain.org data, are there any particular traits on symptoms such as latency period or virulence that correspond to the phylogenetic patterns?
No, I don't think so. I think we currently have no indication that any of the genetic variation that we see in the virus has an effect on how it behaves in a patient. That's not to say that it's not there, but we haven't seen that. And I think whenever it looks like it's there, because like the disease seems like particularly aggressive in Italy, and then people start thinking like, oh, maybe the virus in Italy is different. I think that is probably unlikely. It's much more likely that other things are happening. For example, the Italian population is much older than the rest of Europe, and people tend to live together much more. So grandma, grandpa live together with the whole family in a single house. And so that is much more likely to explain the difference between Italy and other places rather than a difference in the virus. Even if it has a few mutations, it's unlikely that these mutations have such an effect.
Thank you. So I've got a question, I think, all the way from Indiana. If we get 100,000 tests a day, who is to be tested? If currently limited to symptomatic and healthcare workers, who comes next? And I think that'd probably be Christina.
Well, I mean, hopefully if we get to 100,000 tests per day, that can be opened up to symptomatic or asymptomatic individuals. It's hard to say at this point because the number of cases could go up and we still may need to limit it to symptomatic individuals. But we are doing testing of asymptomatic individuals. We do plan to do a surveillance study to see out there whether you're symptomatic or not, if you do have traces of this virus in you using the PCR test. So in regards to the question, it really depends on the situation, but we do hope to test asymptomatic individuals through a surveillance program soon.
Thank
and
you.
then from a health disparity perspective, there's been reports that it's physician bias that decides who gets tested and not. And so we need to make sure that our students who are pre-med, pre-health, pre-nursing, recognize that all people deserve testing, not just, you know, your more affluent patients, because there was an article on that.
Yeah. And so I see a comment from one of our faculty members who's not on the panel, Dr. Wilfred Dinniklaw, and about the CDC. And he says people in the CDC are self-determined and are still trying to work within the CDC guidelines. And in his view, if we don't continue to adhere to CDC guidelines, rural America will be in big trouble. And so that's an opinion from another scientist in the department. Let's talk about, we got one more question here before the next set of panelists come up. But what do we know about herd immunity, if you get immunity, if you've had it, and will the vaccine confer immunity, you know, in the future? Will it be just for this year or will it be on down the road? Anybody know anything about that? Okay, sorry, you're a little bit outside our area of expertise here.
Well, the test has to be done.
I'll try to address that a little. When you compare what's known about the other coronaviruses, they all tend to confer something like 1 to 3 years of immunity. And that includes some of the cold viruses that circulate around. We all get relative immunity to one strain, and then the next season, another strain is circulating. So there's a possibility that that will be what eventually happens with COVID-19 as well.
Okay, thank you, Mike. So at this point, we're going to turn to our second batch of panelists. And Kaho, are you ready for us?
But I'll also encourage the panelists to answer questions online in the Q&A while others speak.
Okay, Kaho, we're ready for you if you're ready for us.
Yes, sure. All right. Okay, so I'm a Kaho Hammer. I'm a postdoc at the Pluny's lab. And again, I study viral evolution, but I'm not an immunologist or any specialist. But because we're waiting for vaccine, we're all waiting for vaccine. And we heard that it may take another year at the shortest. And I wonder why it takes so long. And we had to talk about it in the lab meeting as well. And I started to learn a little bit about the vaccine, and I wanted to share what I learned so far with you guys. So I will briefly talk about the types of viral vaccines. And first is the live attenuated vaccines, and which are made of weakened or altered viruses that will not cause illness. Live attenuated vaccines elicit a strongest and long-lasting response. The second is the inactivated vaccine. They are made of viruses that are inactivated by heat or chemicals, which destroy the virus's ability to replicate. Because they can't replicate, this type of vaccine tend to provide shorter length of protection than live vaccines, and it's more likely to require boosters. Third type is the subunit vaccines, which contain only specific parts of the target virus that elicit an immune response. They can be made of isolated proteins or using vector viruses that express target proteins. And the last type is DNA RNA vaccines, which consist of only DNA or RNA that encodes particular antigens. The advantages of these vaccines include lower cost, easy to manufacture, and DNA is also stable compared to proteins. But challenges include that it's not provoking strong immune responses at this point. So several DNA vaccines are available for veterinary use, but currently no DNA or RNA vaccines have been approved for human use. So as of mid-April, there are 115 vaccines candidates available globally, and which are shown in this modified figure from Nature article. And you can see that the many vaccines are being developed, currently developed as a subunit vaccine or DNA RNA vaccines for speediness. So what are the challenges involved in this vaccine development? Let's see. So currently, many are based on the spike protein of coronavirus. Spike protein mediates the entry into the host cells and are considered promising immunogen. But the key issue is if they generate enough T cell responses. Spike protein will definitely give antibody response, but antibody response alone is not enough. And the spike protein, along with other internal proteins, are considered to be needed to direct T cell, balance T cell responses, and determine the best antigen to stimulate the immune response is a tricky and long it takes time to achieve that. Also, to elicit strong immune response, many requires additional ingredient called adjuvant, which requires another optimization steps. And lastly, these subunit and DNA vaccines likely lead to responses that way more quickly. So there's another step to be worked on. So and then also the preclinical experience with the vaccine development of SARS-1 and MERS have also raised the immunopathological concerns, since antibody-dependent enhancement and vaccine-dependent damage are both reported from these studies. Antibody-dependent enhancement occurs when antibodies facilitates virus entry into host cells, leading to increased infectivity rather than neutralize them. And vaccine-dependent damage in SARS-1 caused abnormality in the lung cells from allergic-like reactions, albeit no symptoms. So with the anti-vaccine movement rising in this country, what we do not need is the bad vaccine that makes the illness worse, and thereby we need to adhere to the strict monitoring. And because of this difficulty, traditional vaccine development actually takes 10 years in average with a high failure rate. But in the midst of pandemic, the COVID-19 vaccines are being developed at an accelerated timeline with overlapping clinical phases. So if we can make these vaccines in next 18 months. That is indeed a very short time, and let's hope that it will get there. And with that, thank you for listening, and I'll share with the beautiful, I don't know, I shouldn't say beautiful, but COVID-19 picture, and I'll take a question later.
Okay, thank you so much, Kaho. We're now going to move to Ravinder Sehgal, who's going to talk about SARS and COVID-19 and how do coronaviruses replicate, which is obviously important for their infectiousness.
Hi, everybody. Can you hear me?
Yeah.
Okay, great. So what is a virus? This is kind of important to understand. What is a virus? It's not really alive. That's something that people are a little confused about. So viruses, you have to think of a virus like this little flash drive that has information on it, and then you stick it in the computer, and then it can tell the computer what to do, right? So it's not really this living thing. So when people ask, how does this virus survive, or how does it live? It's not really alive. It's just a little protein basket with little pieces of RNA inside. So let's go a little bit into this. I found a New York Times article which was really interesting. I have the link for it on the bottom. It talks about all the different proteins that are on the virus and the different aspects of the viral proteins and the RNA. So remember that there's DNA and there's RNA. This virus is an RNA virus. And so in this little depiction here, you see the pink stuff in the middle. That's the RNA, well, the yellow one with the pink stuff around it. That's another protein surrounding the RNA. And then you got this shell that's made out of lipids, and then you got other proteins on the outside. The one in the blue is the spike protein that we were just hearing about. Okay, so again, this is a large envelope, positive sense, single-stranded RNA. So if you've ever learned any molecular biology, you know that RNA gets translated in your cells to become proteins, right? And so this RNA actually acts just like a messenger RNA. It's positive sense, so it gets into your cell and starts to make copies of proteins instantly. And so this is a little electron micrograph of a coronavirus, and it's called a coronavirus because it's got like these little spike proteins sticking out of it, and that looks like a corona on the sun. So that's why they call it a coronavirus. All right, so here's a little bit more. The spike protein we just heard about is on the outside of this virus. It binds to a protein called ACE2, which is angiotensin converting enzyme 2, which is on a lot of your cells. It's on your lung cells, arteries, heart, kidneys, intestines. It's important for blood pressure regulation. That's the normal function of ACE2. It basically reduces your blood pressure. All right, so that gets the spike protein binds to that ACE2 thing. Then there's another protease that helps cleave it, and then it gets into your cell by endocytosis. I'll show that in the next picture. But then there's this RNA polymerase. So you remember that RNA has to be converted to another negative sense RNA in order to be made into more RNAs. And so it has this RNA polymerase, RNA-dependent RNA polymerase. Okay, and then those start to get translated and making more proteins. One of them is called 3A, and that causes cell death through different complicated pathways. One of them is MAP kinase pathway. So basically this virus makes as many as 29 proteins which carry out the range of jobs from making copies of the coronavirus to suppressing the body's immune responses. There's a lot of different things that these little tiny RNA can do. There's 29 proteins. That's not many proteins at all. You know, compared to a human, there's, you know, thousands and thousands and millions of proteins. So this is just 29 proteins, and it's amazing that we still don't understand what these 29 proteins actually do. So
1 minute.
1 minute, S protein binds to the receptor. We got the translation of the polymerase, transcription of the positive to a negative. Then we get some more of these proteins being made. And here's a depiction of that whole thing. The ACE receptor fusion gets in. These RNA positive is made into RNA negative, and that's translated, made into more positives. And then it goes to the ER, goes to the Golgi, and it starts to make thousands and thousands of these viruses that are released. Okay, so that is the main replication cycle of the RNA virus. Remember that it's a positive sense RNA, so it already has like a poly A tail and a methyl cap, so it's all ready to go. It just gets inside your cell and starts making proteins immediately. It's about 80% identical to the SARS of 2003. Remember in 2003 where there was a SARS that came from bats and it went into humans, and that is about 80% identical. There's a few differences in the S protein that are between COVID-19 and SARS. But now remember also that MERS bound to another receptor called DPP4. And so this one is binding only to ACE2. So we have a lot of knowledge about what it's binding to and how this is all working in the cell. Now there's a lot of questions. Where did it come from? I study emerging diseases. And so we think that it came from a bat, right? And it probably, there's 96% identical to a bat that they found in some Chinese villages. So we believe that it came from a bat. There's some talk about it going through a pangolin, which is a kind of armadillo-like animal. But at this point, I think the best evidence is if we're coming from a bat. And then it spread to humans. And this is exactly what happened with SARS from 2003. The MERS went through a camel, so it's a little bit different and had a different receptor. So I think that's a little bit about the information. Now, one thing that we've heard about the vaccines is we have to know, first of all, if we do get immunity to these viruses. If there's no immunity to the viruses, then the vaccines will never work. So at this point, we do believe that there is good immunity and that people who do get infected with this virus will actually be immune to other infections. That's why we believe a vaccine will work. Great, I'll take any questions in the question session. Thanks.
Okay, thank you, Ravinder. Next up is Erica Sanchez, who's one of our lovely junior faculty members, coming to us from Texas.
Hello, can you hear me?
Mm-hmm. You're on.
Okay, I'm sharing my screen. And you can see my screen?
Yes.
Great. So thank you, Laura, and thank you everyone for attending today, and thank you for including me. I thought it would be a great idea to share a little bit about what is happening in our department regarding incorporating the discussion of what's happening, COVID-19 and the virus, SARS-CoV-2, into our classrooms, into discussions and implementing activities around better understanding the virus, but also tapping into a lot of the research being done that's fast, being published very rapidly. There's a lot of information coming at us. There's a lot of different versions of information coming at us from science, from non-science sources. And so I wanted to acknowledge a lot of what our department has been implementing into the classroom, but also resources and open a discussion about resources that you can use as a studied scientist that you can rely on, and to talk about where to get information and get information that you trust. So I am teaching general virology this semester, if you aren't familiar with that. And so for me, it made a lot of sense as the pandemic started to become more and more relevant to the US and to become a pandemic, to implement discussions into my classroom. And so that seems very obvious to a lot of people. But in reaching out to my colleagues, I was really excited to find that discussions on COVID-19 and the virus that causes this disease have been being incorporated into lots of classrooms. And so in reaching out to folks, myself and others have decided to modify or revise some of our original plans. So in addition to modifying and revising our plans and going online to work with all of these students online in all the classrooms, a lot of us thought it would be a great idea to include primary literature. So tap into that very rapidly published work that is coming up in scientific databases. And so if you haven't talked about that in some of your classes, you might be able to ask questions now about where can I find this literature? Is it available to me? You can use repositories like PubMed or bioRxives, which are a lot of the newly published material is showing up on, and we can discuss more about that. But I thought it was great that several classes, several faculty members reached out to me to say that they actually replaced their planned research literature reviews with a very recent publication on the pandemic virus. Also, folks in laboratory classes are actually discussing the testing, the actual ways in which we're testing using that qPCR that was discussed earlier, or antibody tests, and what do those actually look like? So faculty who are in molecular biology labs are discussing this with students, which I think is fantastic, to understand what it means when someone is being tested, whether they're positive or not. A lot of folks are inviting people into the Zoom classroom. I actually had a guest lecturer who was planning to talk about dengue virus with my class, but very awesomely agreed to talk about coronavirus. He works at UCSF, and his lab has completely converted to studying the virus. So this is a time when a lot of labs, especially at large research institutions, have completely switched their focus to researching and understanding this virus specifically. So it was really great to present a researcher who's right there in the mix, and have my students and other students ask questions of that person, the expert, the local expert. Other folks are incorporating online labs. I know we've been removed from campus, but a lot of people have worked hard to include videos, video themselves doing the labs, and to encourage the listening of podcasts. So there are a lot of really cool podcasts. I know there's a This Week in Virology, which I can type in to one of the chat boxes after. And there's another one that I know was good for an evolution class. Jaime Chavez has talked about spillover, another great podcast out there to listen to. We have a little more time possibly to do that. And finally, people have also been discussing the disease ecology, viral phylogenetics, and genetics of the actual virus itself, to incorporate that into the class where we're learning basic human genetics, or how to understand evolution of different species. We can talk about the virus as an example. Folks have been talking about the physiology of the disease. So understanding how the virus is impacting our basic biology can actually help in learning about basic biology. So rather than just memorizing, certain parts of a pathway, some of our faculty, like Jennifer Breckler, has been talking about cardiovascular diseases or diseases of the lungs with using the virus and what we understand about it as an example. And then also very importantly, as Letty Marquez Nagana shared with us today, talking about the community impact and the community service learning activities that we can implement into class, that she has very much implemented into her class. And our ability to communicate with the public as trained scientists and learning scientists. So if we can learn through activities like this and by implementing discussions in our classroom, I think it's great that we can then represent an informed person for our communities, whatever community that you are in, or multiple communities.
1 minute.
Thank you. Engaging students also on Twitter, or as we've seen, Dr. Pluni Pennings, who has been amazingly developing videos to share with folks on evolution and the spread of the virus and flattening the curve. I think it's been amazing to see faculty members actually outside of the classroom, in addition to what they're teaching students, to also be teaching the community directly. And so I really just wanted to take this time to honor that. I'm also, while I am a virologist and a cell biologist, I'm not a coronavirus expert. We didn't have coronavirus on the docket for this semester, but we did a lot of work to incorporate and implement it. And it was nice to see I was in good company. So I think we just want to acknowledge everyone in the department who's participating in bringing these conversations to the forefront, even though we've been greatly altered in the way we teach this semester. So I want to take the time to thank everyone who emailed me, but also to the instructors who are also engaging students who I haven't heard about how they're doing that yet. I'd love to hear from you. And of course, to the panelists today who are running this wonderful discussion for a couple hundred students right now, which I'm really grateful to be a part of. Thank you.
Thanks so much, Erica. This is great to see all of our instructors really incorporating very relevant research into their classrooms. Mike Goldman is next, and I think in these times it's really important to think about what things will look like after coronavirus. And I guess I kind of think of this as a massive wildfire, but at the end, you're going to have new things sprouting and growing again. And how can we actually leverage some of this to make sure that things look better on the other end? So Mike is going to tell us about the post-pandemic world.
So thanks, and my name's Mike Goldman, and I study human genetics. I want to thank you all for being here for this conversation, and thank Laura for organizing this virtual and valuable event. So let's be clear about one thing. COVID-19 is in fact a big deal. It's a once-in-a-century pandemic that's sweeping the whole world, and it is one for which even our wealthiest nations, like us, have found themselves painfully unprepared. It's costing precious lives, and it's blistering the world economy. It's going to rewrite history. While the immediate problems might linger for years, I want to look a little further out, perhaps 3 to 5 years from now. And at least for the students who are listening, 80% or more of your life is going to be lived post-COVID. My contention is that your lives will actually be better for having gone through this kind of cataclysm. But it depends on our learning the lessons of the pandemic and seeing to it that no one forgets those lessons. And biology students have a special obligation and a special opportunity. As a direct result of this pandemic, we will never again be able to elect a president who has a 4th grade vocabulary and a self-centered, xenophobic view of the world. More than ever in my lifetime, we realize that the role of government is to protect the people. And we'll want the smartest, most principled leaders, the most global and long vision, not the guy who graduates at the bottom of the business school class and recommends that people drink bleach. We won't tolerate the failure of insight, preparedness, and leadership again. We will engage more effectively with the most significant problems of our day, such as climate change. We know that the contraction of habitats and the inappropriate juxtaposition of the natural and the anthropic worlds, where humans encroach upon trade and trade in wildlife, is where viruses like SARS-CoV-2 leap from their natural host species to humans. At the present rates of intrusion and climate change, this kind of previously rare event, which we haven't seen in 100 years, will happen more frequently, just as we've seen an exaggeration of extremes in weather conditions. We have to insist that our leaders deal with issues like climate change. We have to understand that when any part of the world is sick, we are too. What I like to refer to as the one health, one world ideal. The essential force for all these changes will be education, with a particular emphasis on science education and critical thinking. The biological sciences will see a surge in interest, and funding education, making it affordable and realistic, will once again become a national priority. Among some other thoughts about the future, I hope that universal single-payer healthcare will finally become a reality. The health system will no longer be one of the world's most expensive and least effective. We'll address many of the inequities that Dr. Marcus Magana spoke of. We'll recognize and encourage healthcare innovation, both corporate and public. We'll invest heavily in public health, something that's been cut by about 25% just since the 2008 recession, will strengthen agencies like WHO, realizing that global health imperative. Science will get better funding and scientists will get more respect. I wager that we'll see enhanced funding of science in general and biology in particular as we recover.
1 minute.
From today's heroes like Tony Fauci, we've learned that science has to lead us out of the most difficult challenge. And we will admit that we don't know where that next challenge is going to come from. It may be a pandemic, it may be something else. Scientific meetings will probably go virtual a little more. We'll have a chance to go to more of them. Like I just went to the Allied Genetics Conference without leaving my chair. We'll see more education taking place online. We'll have to improve the sanitation and up our game in maintenance, even in buildings like Hensel Hall. We'll see lots more of telemedicine. So I think we can see the light at the end of the tunnel. And it's just beaming one health, one world post-COVID. Thanks.
Thank you for that, Mike. I like your view very much. And I want to emphasize to everybody that we're in a really historic situation, and it's really devastating to a lot of communities. But the one thing that we do know is this is temporary. It may be a long temporary, but it is temporary. And, you know, it feels like an eternity when we have sheltering in place extended another month, but this is temporary. So since you brought up kind of the post-pandemic world and people thinking about climate change, we had a question, Mike, about will global warming, and this may go to Ravinder too, will global warming actually affect the spread of COVID-19? Or maybe you want to speak to other things, Ravinder. So Mike first, do you have any sense of the connection?
I certainly think global warming is taking animals out of their natural habitats and forcing them to encounter humans in ways they didn't before. As far as a specific effect on the COVID-19, I don't know, but there certainly will be a COVID 2050 and a COVID 2070, and I think we'll see those if we don't do something to reverse global warming. We'll see these issues arising more and more frequently.
I would say more than global climate change is the destruction of the rainforest and these areas where these bats and other animals live. We got Ebola from bats, we got SARS from bats, we got hantavirus from bats. We get a lot of mosquitoes spread a lot of diseases. So, you know, global climate change will reduce our numbers of tropical rainforests and also bring people into more contact with these animals that can spread these viruses and diseases. So I think we really have to be careful about exploiting our environment and making sure that we're very careful about people coming into contact with animals that might be spreading diseases. I hope there's a lot more research into this than that.
So Ravinder, you mentioned all these diseases that were propagated through bats. Why bats? What's special about bats?
Interesting. They don't have the same type of immunity to diseases that we do. So bats, because they've evolved to fly, they put a lot of their energy into flight, and their food basically goes to flight. They don't have a very strong developed immune system. And so because of that, lots of these viruses survive in bats and they proliferate in bats. And so for that reason, the bats are really good vectors for these types of viruses like Ebola and SARS and some other ones, hantavirus, many, many viruses. So we believe that bats are a super spreader in terms of types of viruses that they can spread.
Thank you.
But at the same time, those bats have co-evolved with many of these viruses, so they don't cause the same amounts of disease in the bats that they do in humans. The real danger in these zoonotic diseases happens when humans interact with the bats and these viruses and these pathogens jump species.
Yes, thank you. And so Ravinder, you mentioned the structure of the coronavirus and that the outer layer is comprised of proteins and lipids. Can you explain to people how washing your hands helps get rid of coronavirus?
Yeah. So lipids obviously are fats. You know how you use soap to wash some oil off of your frying pan? It's the same thing. It dissolves, it goes through and dissolves the lipid bilayer. So very simply, using ethanol or soap will basically rupture the virus and kill it. It's not alive to say, but it will destroy that virus quite simply because of the lipid bilayer. Yeah, thanks.
Thank you. And so maybe Kaho, somebody's asked about thoughts on using interferon alpha 2b as a form of treatment. Do you know anything about that?
I have not read about. Oh, who was going to talk about
Kaho.
that? Okay.
Oh, you can answer too, because I do not know anything about it. So unfortunately, that's more to the go to the doctor question. What I heard so far, listening to the podcast and all that, it's that yes, certain interferon helps, but it depends on when they administer. So there were some studies that when you administer the first week, it worked before it got too bad. But once it got spread, it didn't help, that kind of information. So I don't know the particularity, but I know there's a lot of things going on, but the doctors are being frustrated because this is the first time for them to dealing with. So it's kind of like they're trying and then seeing what comes out, and there's a lot of work going on there.
So,
and the problem,
what an interferon does, interferons basically are messages. So if a cell gets infected with a virus, it sends out this signal called interferon to other neighboring cells, telling them to be careful. And so it's like this little message that tells cells that there's something dangerous happening and they have to prepare for it. So that's what an interferon does. So I can imagine that adding interferons would help to alleviate and make the cells aware that something bad's happening to them. That's basically what an interferon does. I'm not sure if it would cure the virus. It wouldn't cure it, but it would basically tell your body that something bad is about to happen.
And so our resident immunologist, Steen Weinstein, just chimed in on the chat saying that in many cases, interferons will actually aggravate the disease because they also probably, he didn't add this, but they probably increase inflammation, and the lungs are already significantly inflamed as well. The real challenge here, and I can speak to this because my wife is actually trying to organize a clinical trial for COVID-19 therapeutics, is that it's very, very difficult to design a proper scientific study in the midst of a pandemic, as you can imagine. So people are trying many, many different things, and they should continue to do so. But getting good quality scientific data that is actionable, that we can actually design a therapy from, is difficult. And we've seen this, for example, with the hydroxychloroquine story. Where the administration of hydroxychloroquine had two effects. First, that it caused an increase in the number of cardiac arrests due to arrhythmias in patients that were being treated, but it also had the secondary consequences of depleting the chloroquine supply for patients with lupus who are now suffering because they can't get access to the medicine. It's depleted the worldwide supply of that medicine. And so there are clear indications that some therapeutics may be useful, but it's difficult to design a proper scientific study. The same thing's happening with ventilators. It looks like the application of ventilators in late stage disease is not actually beneficial. So people are starting to look at when it's best to use those treatments and those interventions within the progression of the disease. The problem is this caught us all by surprise.
Thank you.
Could I return to a point that Ravinder made a little bit earlier? He mentioned that hantaviruses were from bats, if I heard him correctly. It's my understanding they're actually from rodents, not from bats.
I think there's different ones. Maybe, yeah, hantavirus, there was, what's the other one? Hantavirus was from rodents, but there was another one that was from bats.
Ebola.
Ebola for sure.
Yeah, filiviruses in general.
Yeah, anyway, there's a bunch of
Yeah,
viruses.
I just didn't want to get.
Thank you for clearing up that. Yeah.
Didn't want to get bats badmouth too much, but
Sorry.
Rabies.
I want us to
Rabies
realize
is
that
also-
that not
Rabies
just
is also.
not just the bats are a source of potential pathogens that could jump to humans as well.
Yeah. And so here we're modeling scientific discourse and people kind of updating each other, which was fantastic. Eric, I had a question for you about, can you share some of the online resources that you discovered about teaching about COVID-19, and where might you share them so that people could find those?
Yes, absolutely. I am taking suggestions on the easiest way to get it dispersed to everyone, but I do have a short list of what I've been using and what I've been hearing other faculty and instructors have been using. I did answer, I think, that question a little bit. I would say a lot of the resources that we learn about as new researchers in the lab, like PubMed, which is a governmental website where publications are submitted. BioArchives is, which is kind of hard to spell unless you know what you're doing. It's spelled oddly. That is a preprint site. And so a lot of the new data is going there as we're not waiting for the same type of review. So if it's strong enough, it's being reported so people can continue working on these questions in BioArchives. And then Google Scholar is another way that a lot of my students like to search for things. Virology journals themselves. I think talking with your PIs on where they publish, if they know things about immunology or even everything to like climate change and the environment, there's a lot of publications. There's actually a lot of research money going towards understanding COVID, not just in human health, but also how it's impacting other areas. And then the last one I suggested was JOVE, J-O-V-E, the Journal of Visual Experiments. And they're usually a subscribing service, so you have to pay, but they're actually providing COVID visual experiments and lessons on videos through June 15th for free. And so I've been encouraging my class to use that as well. I'm not sure if I can make a list and give it to you, Laura, and we can post.
Sure, we can figure out how to do that. We'll be posting the recording of this meeting as well somewhere on the department website.
Great.
So I've got a question. Thank you, Erica. I've got a question from Karina Nielsen, who's the director of the Estuary and Ocean Science Center in Tiburon. And she's wondering about the biological mechanisms of so-called super spreaders. Is it just that they're asymptomatic, or are they producing and releasing a lot more virus than other individuals as well? Does anybody know about those?
Well, some of the super spreaders are actually people who have been close to lots of other people. So when they start to figure out, you know, how many infections have arisen from this one person, for most of them, they're not passing it on to anyone, or they're passing it on to one person. But for some of them, they've, let's say, been in a big round table at a restaurant, and you can easily count all 16 people they've spread it to. So that's at least a part of super spreading. But one of the biggest issues with COVID-19 is the fact that so many people are able to spread it without themselves showing symptoms, and that was not true of, let's say, SARS-CoV-1.
Thank you. We got a question here about, are there nations that have recovered completely, and what protocols are they using to make this happen? And so maybe somebody knows, I saw a report today that New Zealand is now COVID-free. Does anybody know about that?
I didn't know about that, that New Zealand is COVID-free, but that's impressive. Of course, they're an island, so it's maybe easier for them to stop new infections. They were also fast in like you know, taking action and making sure it didn't get started too badly. I think several of the Asian countries are doing really well, like China and South Korea and Japan, but they are pretty aggressive in their rules, and also they are very well organized. Like they have a federal government that has that makes sure that everybody gets tested, all the contacts get traced, like in a way that currently in the United States is far from happening.
If you compare our contact tracing idea of 150 hires to do that in San Francisco, what Wuhan deployed at the height of their situation was something like 1,800 teams of 5 to work as tracers.
Yeah, thank you. And so I think we've got time for one more question. And the question is this, that this participant's heard that one of the main pathways of COVID-19 fatalities is due to cytokine storms. Is that true? And are there other pathways that cause fatalities? Can anybody speak to that? I know it's not quite up your alleyway.
I know just a little bit about cytokine storms, and this is true that the COVID-19 does have activation of many, many cytokines. And so that could release the cytokine storm, which is basically a lot of molecules that are signaling a lot of inflammation, a lot of activation of white blood cells in many different aspects. So I do believe that the cytokine storm is an important pathway to the pathogenesis of COVID. I don't know if there's any other pathways.
Okay, thank you so much. So at this point, I'd really like to thank all of our participants for coming today. This was our first departmental webinar, and we had over 259 participants, which is just fantastic. I think it really speaks to our desire to come together as a community to respond to the science of what's going on around us. I'd really like to thank our 8 panelists who took their time out today to prepare a presentation for you and to answer your questions. And I hope that what you'll realize is that we're here and we're around and we can answer questions anytime. You don't have to wait for a webinar to ask your questions. And so there's lots of resources, as Erica pointed out, for people to do their own investigations. I highly recommend the Johns Hopkins site on that has data about COVID virus. But, you know, we're scientists and we're curious and we're always going to be asking questions, but we're all here and we're interested in engaging with you about these important things. And as always, we're going to go through some hard times here, but as a department, as a community, as a country, together we will shine brighter. And so I guess the one thing I would ask for the participants who are still here, if you have any feedback on this panel presentation or what you liked, what would make it better, go ahead and type it into the Q&A now so that we have some sort of feedback about how to do this better next time and what you liked so that we know what to keep and whether you'd like to do it again. So anything you can type in would be great. And at this point, we will sign off, but please go ahead and type in